The reconstitution and fusion protocol detailed here is an efficient way to study membrane-bound proteins in a model lipid bilayer and lipid mixing by fusion proteins. Here we describe a detergent-assisted reconstitution of recombinant Drosophila atlastin, an ER fusion protein, into preformed phosphatidylcholine liposomes. Fusion of atlastin proteoliposomes is then measured by a FRET-based lipid-mixing assay. One of the key advantages of detergent-assisted reconstitution is that the protein in detergent is not exposed to drying or solvent. We also show here that reconstitution and orientation of atlastin is very efficient. Another advantage of the FRET-based lipid mixing assay is that the liposomes do not need to be loaded with any dye, and there's no need for any extra dialysis steps.
To begin this procedure, prepare the lipid mix stocks in chloroform as outlined in the text protocol. Add 1 microcurie per milliliter of DPPC to the lipid mixes, making sure to reserve at least 8 microliters of this stock. Transfer the desired amount of the lipid mixes into flint glass tubes. Then dry the lipid mixes under a gentle stream of nitrogen gas for approximately 10 minutes until no more chloroform is visible. Dry the resulting lipid film further in a desiccator by vacuuming for 30 minutes. After this, add enough A100 with 10% glycerol, 2 mmol two-mercaptoethanol, and 1 mmol EDTA to the lipid film to bring the concentration back to 10 mmol. Resuspend the lipid film by vortexing lightly for 15 minutes at room temperature. Freeze the hydrated lipids in liquid nitrogen. To thaw the liposomes, let the solution sit at